In class we talked about how the gradient, the proton gradient, is the driving force for the synthesis of ATP. In this video I want to talk about how that proton gradient is created. We said in class that the proton gradient is generated by the energy that comes from the breakdown of fuel molecules such as glucose and fat, and we haven't looked at how fat contributes yet. We've really focused on the glucose so far, but that the energy of their breakdown is conserved in NADH, the electrons that's carried by NADH, which was generated in the citric acid cycle. We want to look at how those electrons generate that proton gradient and how that proton gradient is used to make ATP. The proton gradient is generated by the passage of electrons through a series of enzymes in the inner mitochondrial membrane, which is here. Those enzymes are called the electron transport chain because they carry electrons, and in so doing, as the electrons are passed from complex to complex, from enzyme complex to enzyme complex, and that's represented by this yellow line. These are the electrons. As the electrons pass from complex to complex, hydrogen ions are pumped across the membrane, and the energy for pumping those hydrogen ions across the membrane comes from the passage of electrons through the complexes. The final electron acceptor is oxygen, and oxygen is combined with those electrons and hydrogen ions to generate water. The whole process is called aerobic respiration. When we think about respiration, we usually think about breathing. So, how our lungs take in oxygen and expel carbon dioxide. This is cellular respiration, but it is related to breathing in oxygen and breathing out carbon dioxide. So as we go through this process, think about the fact that the reason why we breathe in oxygen is so that the oxygen is present in our cells to accept the electrons at the end of the electron transport chain. Before we talk about the details of the electron transport chain, let's consider the reason why the electrons pass from complex to complex. To do this, we have to learn the term redox potential. And the redox potential is a measure of the tendency or the ability of a chemical species, in this case the complexes, the enzyme complexes, to acquire electrons and to thereby be reduced. So the electron transport chain is a series of redox reactions where electrons are transferred from molecules of lower redox potential to molecules of higher redox potential in oxidation, reduction, or redox reactions. Each of these reactions has a negative free energy change and therefore releases energy. The energy is used to pump hydrogen ions across the inner mitochondrial membrane into the intermembrane space, so from the matrix out to the intermembrane space. The final electron acceptor, so the molecule with the highest redox potential, is oxygen. On this graph, we've depicted the free energy that's released per electron on this axis, and on this axis we have the redox potential. It goes from a negative to a positive, so higher redox potential is on the bottom. The NADH has the lowest redox potential of any of these molecules, so it gives up its electrons to the first complex, the first enzyme complex in the chain. Passes those electrons off because that complex has a higher redox potential or a higher ability to bind electrons. That complex passes the electrons onto a carrier molecule called ubiquinone, or abbreviated as Q. Q passes the electrons to the next complex in the chain, which passes them to cytochrome C, another small carrier molecule. Cytochrome C passes the electrons to the final complex, which passes them on to oxygen. So we have a series of enzyme complexes linked by small carrier molecules, each one with a higher redox potential than the previous one, so with a higher ability to bind electrons. In the electron transport chain, we have 4 complexes. The first complex accepts electrons from NADH. The electrons pass through a series of iron-sulfur centers through the complex and are passed on to ubiquinone, or Q. As they pass through complex 1, the energy that's released from their passing through is used to pump 4 hydrogen ions across the complex, across the membrane, through the complex, across the membrane, into the intermembrane space. Q passes those electrons on to complex 3. Q has another source of electrons, and that is complex 2, which is also called succinate dehydrogenase. We talked about succinate dehydrogenase today as being the unique enzyme in the citric acid cycle that is embedded in the inner mitochondrial membrane and that is part of the electron transport chain. So electrons come in to complex 2, also known as succinate dehydrogenase, from FADH2, which was generated by the enzyme, and they're passed through succinate dehydrogenase also to Q. So Q has 2 sources of electrons, complex 1 and complex 2. However, complex 2 does not pump any electrons through the membrane. Q passes its electrons onto complex 3. They pass through complex 3, pumping 4 hydrogen ions across the membrane. The electrons get passed to another carrier molecule called cytochrome C, which pass the electrons to complex 4, the final enzyme complex in the electron transport chain. As the electrons pass through complex 4, the energy that's released from their moving through the complex is used to pump 2 hydrogen ions across the membrane. The electrons are finally passed to oxygen, which combines with some hydrogen ions to form water. Ubiquinone, the first small carrier molecule, which receives its electrons from complex 1 and complex 2, is also called coenzyme Q, CoQ, or just Q. Ubiquinone is a fat, it's a lipid, and as a lipid it can actually float inside the membrane. You can buy CoQ as a supplement to give you a so-called energy boost. So here's Q with that lipid component that carries the electrons into complex 3. Cytochrome C is a protein. It's a peripheral protein, which means that it's loosely attached to the inner mitochondrial membrane. It's a hemoprotein, which we talked about today in class. Those are proteins that have succinyl-CoA as a precursor. This is the structure of a heme group. It has an iron center, and iron is an excellent electron binder. This is what the heme group looks like as part of a protein. So the electrons are passed via the complexes and these small carrier molecules. Q is a lipid, cytochrome C a protein, to the final electron acceptor, oxygen. The end result is the formation of a proton gradient, which leads to chemiosmosis. In other words, an electrochemical gradient. It's also called the proton motive force, which basically means that it's a potential energy formed by a proton gradient. This proton gradient has electrical, concentration, and pH components. It's a very powerful gradient. Again, remember our analogy to a hydroelectric dam. And we saw in class that the passage of water through that dam can be controlled in such a way that it turns a turbine and can be used to make energy, to convert it to electrical energy in the case of the hydroelectric dam. In the case of the mitochondrion, the gradient can also be used to convert energy from the potential energy of the gradient into another form of energy, in this case the energy needed to make a molecule of ATP. In this figure we see the enzyme that generates ATP. It's called ATP synthase. ATP synthase is a enzyme made up of many different peptide subunits. In this diagram, this is the inner mitochondrial membrane. On this side, on the outside here, this is the intermembrane space. And this part extends into the mitochondrial matrix. So the gradient has a high hydrogen ion concentration on this side and a low hydrogen ion concentration on this side. And this enzyme allows for the controlled passage of hydrogen ions down their electrochemical gradient as they pass through this, what's called the FO portion of the ATP synthase. They cause the portion that's inside the cell to rotate, and as they rotate, the energy of that rotation is used to make a molecule of ATP. For each ATP synthesized, 3 hydrogen ions pass through the membrane via the ATP synthase enzyme. Here's another representation of that enzyme. This shows a ribbon diagram of the enzyme just to give you an idea of the 3-dimensional structure of the enzyme. This is the part that's embedded in the membrane, and this is the part that extends into the mitochondrial matrix and rotates to forms ATP by a process called rotational catalysis. This ATP synthase is sometimes referred to as complex 5 of the electron transport chain. The function then of the ATP synthase is to use the electrochemical gradient to drive phosphorylation of ADP to form ATP.